Recording. So I welcome everyone for being here today. We really appreciate that. And today we have Dr. Sharon Batanin, who will show us how machine learning is used as an efficient early warning sign for the emergence of new persistent variants. So Sharon studied physics at the University of Lyon in France, and his research includes high energy particles physics, and especially the Higgs sector. He recently joined a unique collaboration between string theorist, particle physicists, lattice physicist, experimentalist, and computer scientist, and later joined virologists and microbiologists to study the COVID-19 pandemic. Shara, you're welcome to present.
Thank you. But first, I would like to thank the organizer for giving me this opportunity. I mean, being able to share what we've been doing with my collaborators is great, and being with all of you is I'm very honored. So I will start by sharing my screen. Okay. Okay, is it working?
Yes.
Perfect.
Yes.
So indeed, I'm in a kind of unique collaboration with amazing people from different frontiers. The majority of them are physicists like me, but also we are working different area of physics. But now recently, we also have a microbiologist, especially Andrea Cimarelli and Maria Luisa Chiusano, that have joined us and help us with all they know. So this collaboration started when the COVID struck. At first I was not in it, and they did a very nice project, and they end up with very nice papers, and I think I will start to present them. So here my plan. So to present you the complex epidemic randomization group. I think it's a nice way to motivate why we need to go to investigate from variants and why we use machine learning. So this is really not my work, but this is the start of our collaboration. I think it's nice. It's a nice thing that I present that to you. So then I will present you the machine learning goal, what we are aiming at, on the first analysis we did, and finally the latest status, which is a time bin analysis, which I think is the most sophisticated one that we have right now. So the complex epidemic randomization group. Everything started, as I told you, when we had the first wave of COVID. And my advisor of my PhD and another collaborator, they were away, and they are very good scientists. And as very good scientists, they ask themselves very scientific questions, like, when can we travel again? That was their main issue at that time. Well, actually, they really started to think scientifically about that, because they were completely under lockdown, and they were trying to know when the wave will be stopped. So they start to think really naively that if you look at the cumulative number of cases, can you see my cursor? I did not ask that before, but
sure, yeah,
perfect. So when you look at the cumulative number of cases along the time, there is two very important points. So there is a point in which the wave starts, and the point in which at the end the pandemic is over. And those points, they are quite special for two reasons. Because what happened before the first point and after the first point is time invariant. And this property of having time invariance and to have this symmetry for us in our field in physics, especially in particle physics, is always very important. We use symmetry to simplify a lot of problems, and that's what they were doing here. And they were saying that this means that those 2 points here in red are fixed points, and at those fixed points, you have, when you arrive at those fixed points, you are in a time invariant theory. And there should be something that is going to link those two fixed points. And they knew about comportamental models, but they just thought that they can do an analogy with what we are doing in physics. And maybe this could have a nice prediction. So we have this renormalization group vocabulary in which we look at this kind of dynamics. So where we have fixed points on some what I call running, you have a quantity that is going to run from one fixed point to the other. And this is also very close to the idea of scale invariance in physics, where scale is the energy, and here it's going to be time. And we study different operators, that those are like quantum operators, the ones that are relevant and going to really help you understand the dynamic and where are your fixed points and how you're going to go from one to the other. And everything is inside what we call a beta function. So the beta function, the simplest beta function, just here we can do something analogous. I will say that I have something I want to study, which is alpha, a function of the time. And I want a beta function with two fixed points, one in 0, so this one, and another one in A. So usually those kind of theory we have, there is no time, there is a scale of energy, but they use this very simple beta function, and they look at alpha not as the usual coupling constant that we have in quantum theory, but as really the cumulative number of infected. They were saying that maybe alpha, if we see it like this, should follow this time equation. So that's what they did. And the different solution you get in those models, logistic function, they look like this. This is very simple, very few parameters, and they just try to fit that with the data that was available. So there was the first wave in Hong Kong for the SARS-2 COVID, I think it was in 2003. So in blue, you can see the data, and in red, the fit they obtained with this function. So the fit is very good. In China, at that time, we were more or less already at the end of the wave. So the fit is not bad. And this is for South Korea. So this was nice, but I mean, we also have data in France and in Europe, in the US. So they tried not to predict because the data we had was really right in the middle of the wave. So with the uncertainties, for example, in Denmark, one of the collaborators is Danish. If they try to fit the first infected cases that were reported, they follow this. They find this kind of behavior at the end. So this 10,000 of cases should be the maximum of cases that the country should go through. The same in the US and the same in the UK. So this was very nice, but I mean, comportamental models are already here. So I mean, why using that? The fit is quite good, and it's very, very, very easy. I mean, there is few parameters, so it's very easy to fit with something like that. But after a few months, something was a bit wrong. Is that usually that is the kind of behavior you expect. I was saying you at the end of the wave, you hit this flat section in which you don't have new cases. But actually, what was observed in all the countries around the world, that you get a linear behavior after the first wave. And this was quite new. And my collaborator, they just called this the Stirling phase, in which the new number of cases each day is constant. And there was no explanation. I mean, in comportamental model, you need to have some parameter that depend on the time to get something like that. So there was no model that was able to predict that phase. But they found a way by still keeping in this renormalization group framework. And the way they did that is with the complex epidemiologic group framework. So they changed a bit the beta function I was using before. I just absorbed some of the quantity like gamma and alpha, they're useless here. What they changed is that here they put they put a delta, which was equal to 0 before. And you see that if you have delta positive, you get two different fixed points. The dynamic should be exactly the same as before. You should go from the fixed point in 0 to the lowest fixed point, which is alpha 1. The things become to be different if you have delta which is negative. In that case, alpha 1 and alpha 2 should be complex. And if you look at that plot, which is a plot of, so here you have the fixed point in 0, here you have the two complex fixed points in the case of delta negative. And the flow of alpha is going to go through between those two fixed points. And so let's look a bit the slice of this 3D curve. In blue, those are the slide. In blue, you have the case in which delta equals 0. So, sorry, I forgot. If you have any question, just interrupt me. No problem. I just forgot to say that at the beginning. So this was the case before that we were studying. So you have two fixed points. Okay. Now, this is the case in which you have delta which is negative but very, very small. And in that case, you get a local maximum. And actually, the fact that you have a local maximum is going to give you a storing phase. So in the case in green and in orange, you see here that you get a phase in which it looks like linear. Delta cannot be too big already. For delta 10 to the minus 2, you are already starting to lose this storing phase. And at some point, you completely lose it. So, I mean, this was, again, a very simple framework and very easy to fit. And it's what they did. So they did, again, the fitting. So for this, you have the data for Italy, France, Spain, Europe, UK, and Germany. In blue are the actual reported cases. In green, it's what they obtained first with the simple epidemic randomization group without any complex fixed points. So you see that everything is flat at the end. And in red, it's what they got with this new beta function on the complex fixed points. So the fit is much better. And you see that now we can accommodate for the storing phase. But this is very good. And this, if you have after you want to ask me some question, I have the value of the parameter that has been chosen. So this is good, but we can also predict maybe a bit more. And a way to predict, because if you just stay here and we let evolve the red function, it will blow up. You don't have a fixed point anymore. They are complex, but you don't have any more real fixed ones. So this should go to infinity. A way to go away from that is to change the beta function for the third time and to add a new fixed point, which this time is gonna be reordered. So you have the 0 fixed point, which is when you have 0 cases. You have the fixed point that's gonna give you the first wave and the storing, and a third fixed point that's gonna end really completely the spread of the pandemic. So for example, this is what, so you see that now the fit is much better. We can have the first wave, the storing phase, and also it seems that the start of the second wave is really better than before. If you look at before, France, here it was going to be too big and we cannot fit with that. But now with this framework, the fit is much better. But this fit is going to predict that the finite number of cases should be around 400,000. And now if you look at the reported cases at the end of the second wave, just took that on the internet, it's around 2.5 million. So something was wrong. Something was completely loosened. There was an effect that was not captured in this beta function. And we realized very soon that this part of the part in which the wave keeps growing was due to the British variant. And the notion of variant was completely absent in this framework. It seems to be that it's the same disease that is spreading, whereas no, it's another variant. So this was a game changing for us. And really, we did not know what to do at that point. We were trying to write a review for physicists about epidemiology. And we were quite puzzled by that. And we wanted to study the impact of variant, but there was some issue. There was no clear identification of the variants in advance. That disease was something that we knew after, but not before. What was called variant, I mean, we needed to wait for WHO to declare that this is due to a variant, this is not due to a variant. I mean, we did not have really the data that we have for the reported cases, which is very easy to fit. But we found that there is a huge database, which is called GISAID, that is containing a lot of sequencing of the COVID on different countries all around the world. Some countries like the UK, they have a lot of sequencing, around 1,000, 10,000 a day. Some other country like France, for example, was not doing the job. We don't know if they're not doing the job, but the data was not available on this website. So we thought, can we do something with that? And I mean, maybe we could just try to regroup the different sequences that are available by some similarity and plot the temporal evolution of that to see if we are able to define our own variant and then study the dynamic, state dynamics. So this is really the start of this machine learning project. We really started that because we wanted to be able to fit something. We wanted to be able to understand the dynamic with our own level. And I mean, all the definition of variant, everything was a black box that we could not really understand. So by finding this database, we just tried. We didn't know where we are going, but we had to try. And this is really the beginning of this machine learning algorithm. But I mean, we are physicists, and none of us was aware of how we can cluster data. And we go through a bit of literature, and we understand that machine learning can help us. So I don't know if the people here are familiar with that. I'm just going to review very quickly this kind of procedure I'm going to use. And I'm going to use here 2D points. So here you can see on the plan. And if I want to regroup them in cluster, I mean, by hand, it's very easy. I can say that those one are very close to each other, and maybe those one could have two groups. But I would like to have a very nice protocol that help me to regroup those points. And this can be done through what we call the tree of proximity. And so I need to build this tree. And this tree is very, very simple to build. So the idea is that I will start to look at the two most closest points here. And actually, it's 2 and 3. That's why I'm going to color 2 and 3. And the height to which I join the point 2 and 3 correspond to the distance, Euclidean distance when in the plan of 2 and 3. Then I will again look for the other closest one. Here it's 5 and 9. And I keep doing that, 6 and 7. Then, I mean, what the closest thing are not the point themselves, but the group of them. If instead of the point, I use the center of gravity, then the closest things is the center of gravity on 5 and 9 and 6 and 7, here and here. So I'm going to join that. And I keep doing that. I keep doing that. Now I have 1 and 4 that enter the game, etc., etc. And each time the height corresponds to the distance. And at the end, I end up with this tree. And now, if I want to have group, I just need to choose a threshold. And the threshold is just a height. If I choose this kind of height, this will cut my tree in 2 branches. And the leaves of those 2 branches are going to be my group. So you see that if I choose this threshold, I will get the 2 groups that seems to be very easily recognizable by hand, but we needed really a mathematical way to define them. I can use another threshold. Same thing, that time I will get 3 because I'm also cutting these branches, so I get 3 groups. And this is exactly this method that we use for our data. But we had to choose a special strategy because we are not in with those 2D points. We have sequencing, so it's a bit different. So how do we compare two sequences? We know that in biology, there are some scores that are defined, but this was not really clear to us what was the motivation to compare those. So we went to something very simple. Sorry, I was getting ahead of me. What do we compare? We are going to compare first the proteins, the spike protein of the viruses. We have access, for example, to the whole DNA of the viruses, but this is quite long. So it's going to take time. And also in the DNA, you can change some stuff that will not change the sequence of protein. So looking at directly to a sequence of protein is more efficient, we thought. And also the spike protein is really the spike that enter in the mechanism between that helps the virus to go to the host cells. So it's really the key of the spread of the virus in a sense. So it's a key protein, and we thought that let's start with that one. And it turned out to be a very good choice. So how now do we compare those two sequences? Sorry, I got ahead before. So I was telling you that there is a score that is defined in biology to compare two sequences, but again, we went from naive stuff. We use what is called the Levenshtein measure. If you give to the Levenshtein measure two sequence or two string, two list of character, the result is the minimum number of operations that you can do to go from one string to the other. And by operation, I mean an insertion, so like a mutation that gives you a new nucleotide in your sequence, or a deletion, one that disappears, or you change, really, that's purely a mutation, you change one of the amino acid. So this is the definition of the Levenshtein measure, and as you see, it's unweighted. I mean, I can change A into a B or A into a C. It's the same thing. It will count as the same. So we choose that, and the last thing we had to choose is how do you compare two groups of sequences? Because before, with our SUD point, I was telling you that I'm going to compare with the center of gravity of the group with the rest of the data. But here, we are not in an Euclidean space, so things are a bit more different. And there are actually different methods to use. There is the single method, in which the distance between two groups A and B is the minimal distance between the constituent of the two groups. There is a complete method, in which it's the maximum distance. You have the average method, in which you just average the distances. And the one we use is the worst distance. So this seems to be very ugly formula, but if I go back to an Euclidean case, you see that this formula will be simply like this, where GA and GB are the center of gravity. Just showing you this, it's not the one I use because we are not in an Euclidean space, but this formula is going to transform like this in an Euclidean space. And I'm just showing you that to understand a bit how the distance is evaluated. So I'm telling you that we use the world method. Actually, we used all of them, and most of them was giving us the same result. We just realized that the world method was the most efficient way to really obtain our cluster. But most of the methods were giving similar results at the end. But now we are just sticking with the world method because it works very, very well. So the results of that. This is what we obtained for England. So England, with the method I was describing to you before, on the threshold of 2000. So here you have the first panel, you have the number of sequencing since January 2020. So you see that, I mean, at sometimes the sequencing can be very high. So after the first wave, it was not very high, but then starting the second wave, they are doing a lot of sequencing. Here you have the frequency of the different clusters that we found. And you see there are 3 colors or 3 clusters. And here I have the 3, so you understand that how those 3 clusters are coming from. With this threshold, I just get those 3. different groups. And if I plot this frequency, I see that there is one which is starting. So here, I mean, we should not pay attention to those points because we have so many low sequencing that this is not really trustable. But we have one main group, and then a second one started and took over, then that disappeared, and at the end, a third one took over again. Now, if we multiply this frequency by the number of cases that we know, this is what we get. So, I mean, what we can see from here that the first wave that was here was caused from one variant. The second wave seems to be caused by the same variant up to the point in which the British variant in blue enters. And actually, I plotted the British variant that is defined biologically here. You see it's in black, but the blue and the black, they match completely. So you're not even seeing the British variant because it's really the blue one. So with our method on this threshold, we're able to see that this is the British variant. And actually, this is the Delta variant here that you're seeing here. And now with a lower threshold, I will get four different groups. And what is interesting here is that now the second wave starts a bit with the same variant as before, but you have another variant that is taking over, the red one here. And this, if you look at the WHO website, this was not named a variant. I mean, no one really paid attention to that, to this kind of sequencing that was spreading. But what we found here is that actually this is one species of sequencing that is a bit different from the other and is the one that is taking over. So if you look like around 70% of the different viruses were in that group at the maximum. So this is one kind of the first result we had. And the issue with that is that, okay, it's very good. We are able to define our own variant. We were able to fit some of the curve with our complex epidemiological randomized group equations. This was nice. But this is an analysis that takes a lot of time. We have a huge amount of data. We need to compute a lot of distancing between all the sequencing. And we very soon understood that, I mean, there is no reason to compare the sequencing of happening here with the one that are here. They're going to be completely different. And each time a wave is going to disappear quite quickly. And if we still trying to run this analysis, I mean, the new data that is going to appear each day is going to be completely dive into the previous data. So that's why we went to this time bin analysis. So the idea of this time bin analysis is that we want to be more sensitive to the latest data. And for that, we're going to divide our data set by month or by week. We could do, I mean, you could choose the interval that you prefer. We just try with those two. And we're going to compare and do the machine learning algorithm to obtain different clusters at the level of each month or each week. Then we need to link the different cluster that we obtain in one specific month to the next one. And to link them, we define two different links. There are the strong links. So those one are going to link two clusters in two different intervals that share the same dominant variant, same dominant sequence. We also having like weak link, if I can say. Those one are gonna join two clusters that are the closest one if you use the distance I was saying you about. They should not be, I mean, the branching links are only in the case in which you don't find any strong links. So just to illustrate a bit, suppose that at the interval T1, I get 3 clusters, the same at T2, same at T3, just taking at an example on T4. And at T2, I'm going to compare the cluster I get with the previous one. And I will see that this cluster and this cluster share the same dominant variant. So I'm doing a strong link. For those two one, they are very close to this cluster. this one is not really close to those ones. So I'm going to do the same between T2 and T3, and then T3 and T4. And I see that I have a chain of strong links that is stable. And this is what now we're going to define as a variant. So before, we were only looking at the big group that we were organizing. Now each week, each month, we have some cluster, and the variance is going to be defined by the cluster that I linked by strong link. And this is exactly again what we did. So this is the result for England again. So this is first here you have for a threshold which is around 200, sorry. This is the chain of link that we obtained. And you see that you have one chain of strong link V0, another one V1 and V2, then you have V3. We can plot again the frequency. You have here the frequency, and here the frequency times the number of cases. And what you realize, this plot is very similar to what I was showing you before. You have V0, that is the original COVID, as I was talking before. Then V1, the one that dominated the second wave in UK, and V2, which is the British variant. We're able to identify it. And finally, V3, the Delta variant that is appearing here. We can do that with a lower threshold. In that case, the variant V1 is going to be divided into V1A, V1B. You can see that. And the same for what we were calling the Delta variant is going to be divided into V3A, that's not really going to dominate, and V3B. That's really going to dominate the spread. And I mean, with those results, we started to think that maybe now we can have an early warning system at the end. We want to be able to very quickly identify, because I can run this analysis in the week I want, so I can run it right now and look at what are the new cluster I get and how they are linked with the past cluster. And maybe with this, we could have an early warning system that will tell us, be careful, this new variant, this new chain of strong links is going to be a variant of concern. And so we have this rule of 3 as an early warning. So the idea is that each time we get 3 different strong links between consecutive cluster, it's going to be very, very dangerous. And we say it's an empiric rule because we get it from all the data we have been analyzing. And each time 3 different links were following one to the other, we had a variant of concern, and WHO declared a few times after that there is a variant of concern. So here, for example, here I was showing you, sorry, if you understand, this was a month-by-month analysis. And if you get this rule by 3, you need 3 months to be able to identify a cluster. I mean, this is a lot. So if you use it by week, it's the same thing. And in red here, you have, for example, those are the number of weeks since 2021. And you see here, this corresponds to the Delta variant. And we can see after week 47 that it's going to be a variant, which is very dangerous. So this is summarized in those different weeks. So the first detection that we know a posteriori of the Delta variant was in week 38. Should be here. But this is the first detection that we know now that the Alpha variant, we know it's very, very dangerous. If now we look at here, the first new cluster that we get that contains that we know of the Delta variant is here. And at week 47, so we have the rule of 3, we should be able to declare that this is a variant of concern. Here, sorry. And if we have been using the monthly analysis, then it's at week 49 that we will be able to say. If you look at here, it's it will be 49 should be here. So, and if you look at when the WHO classify the alpha variant as a variant of concern, sorry, I'm misleading between alpha variant and alpha variant is here. Sorry, it's V2. So week 49 should be here. So if we look at WHO, the classification was done in late December, whereas with this weekly analysis, we could say that there is a dungeon variant in the middle of November, so much earlier. So this is what we are doing now. We're trying to, this program that we are using for the global analysis, it's worked perfectly. This is finished. Now we want to fastest the process of the time bin analysis, and I hope that for Christmas this will be done. And the idea is really now to develop a website that so that we have the help of Sanofi to do that. That we do a weekly analysis. We don't know still if we're going to do that just in Europe or in just France, but every week we want to have a website that's going to do this analysis and tell people that if there is something to be concerned or not. So we are still trying to use that. We now in few weeks we will know about the Omicron, and we're going to investigate that. And the last thing that I would like to say is that this can be, I mean, this machine learning, the first tool that what I was explaining to you is about this early warning, which is great. It could be great if if this can be confined and the performance can be of the algorithm is better understood and we can trust it. This will be great. Another thing is that it's also we are opening this black box of variant that we did not understand, and by clustering all of the different sequencing with that method, and we are just using the spike protein because biologists are using different proteins to that. Here we see that the spike protein is really the protein that is the motor of the mutation or the motor of the spread. And this maybe now that we can separate different cluster, different variant that were not considered as variant of concern, but still are present on creating wave. We hope that we can maybe better understand the dynamic between those variants, the evolution, how a variant is transformed during this life to better control the pandemics in the future. So thank you for listening to me. And if you have any question, I'd be very happy to answer it.
Wow, thank you so much, Sharon. That was a great presentation. I found it very intriguing and new to me. So I have a few questions, but let me go one by one, not to confuse myself. And I'm sure some other people have questions as well. But the first question I will ask is, if the variant is not, if the new variant is not dominant, so is the algorithm able to detect the new variant?
So we have some. So I did not went into the whole details, but we always prune the data we have access on the side. So we have a first pruning on for this machine learning algorithm, the cluster we are retaining. As opposed to each time those clusters they have some size, and we want the size of those clusters to be at least 1% of the sequence we have at that time interval. So if you have a variant that appear but not spread a lot and it is less than 1% of the whole sequencing, then we will not detect it. But if it starts to be important and it goes above 1%, then we start to detect.
So the 1%, was it assumed based on?
So we actually I don't have other graph here. We had also some, we played here for the alpha variant with different threshold and different, I mean, lower threshold of this 1%. We played a bit I mean, the issue if you have less than 1% is that you're gonna have a lot of clusters that are gonna appear. I mean, with too many clusters, it's getting difficult to really put your hand in the one that are dominant, and the one that are dominant are the ones that are big. So it's a way to throw away some data that is still not very useful. But if a variant is going to be dangerous, it has to be over 1% of the sequencing. So this, I mean, also when I was talking about the performance of the algorithm, those are stuff that we need to be sure how to adjust the threshold, but also this 1% rule.
Good. Yeah, so my next question before I open up to other people is, first thing I would like to point out before the next question is, I assume that the British variant is alpha, right? If I'm right?
Yes.
So I'm just wondering, like, maybe it's better to call it alpha variant or instead of British variant.
Yeah, sorry. It's, we said that because it appeared in the UK the first time, and
yeah.
It's alpha variant.
Just to quick, hang on a minute. I thought the Kent strain was beta, wasn't it? Or was it alpha? Anyway, whatever it is, I double check which the new year it was. I can't remember either, but I for some reason my brain is telling me it was the beta variant. I might be wrong.
I think it was alpha.
Was it? Okay. I thought alpha was the strain responsible for the first wave.
Yeah, it's also something that bothers me each time that I'm thinking about alpha, because alpha seems to be the first, but I think, yeah, it's really alpha that the name for this first big variant that was identified.
Yeah, I think beta, and I think the Kent strain was named British, but in Britain we called it Kent. Was I think was beta. Double check, just to make it's right in terms of naming. But I think the entire WHO thing was to try and stop geographic, the naming thing to stop this geographic association.
Okay.
There's an idea of possibly be blaming the country it's from.
Yeah, you're right. And I will also need to double check the alpha beta. I need to be sure. You're right.
Good. Then just quickly, the last one is, I'm just wondering if you want to share how this algorithm has been helpful towards detecting the Omicron. Do you have an idea?
But right now the Omicron is not really have a big presence in the data set. I mean, we are not talking about Omicron, but the data needs to be on GISAID, and GISAID always has a bit delay. So I think the first detection of Omicron was like a few weeks ago. So we always have this delay of getting the data on GISAID, and we need them to be sufficiently present with those 1% rule, etc. So right now we cannot say anything about the Omicron, but I mean, again, here people are saying that Omicron is going to be dangerous, but we still don't have any epidemiological data saying that there is a wave of Omicron. We know there are cases of Omicron around the world, but nothing to quantify the effect of the Omicron.
So so right now the algorithm is probably working in retrospect and not like detecting the future variant or so. Like, I mean, you're using the algorithm to test the previous variant and not for the maybe upcoming or recent variant.
What we have is not something that is called variant by WHO. We just have some cluster, and with this rule of 3, we see that they start to becoming persistent cluster, and this is how we can say that they might be dangerous. So this is how we can really prevent. I mean, all we are doing, all the whole analysis, of course, is on the past data, but with this refined analysis, we are able really to maybe detect in advance what can be a dangerous variant.
Good. Thank you so much. Martin, your hand is raised.
I'm not sure if this was answered because of the fact that there was a bit of a problem with my connection. Your method seems to be extremely relying on sequencing data. I mean, like you said in the talk, France was a bit lacking compared to the UK, but I think the UK was very ahead in terms of what other countries were doing. I mean, an Omicron originated in Southern Africa, not just specifically South Africa. No one's really, from what I understand, no one's really sure, and many Southern African nations do not have the sequencing capacity of, say, the UK or even France or anywhere else for that matter.
Yeah.
Any Western nations for that matter. How does your method going to be able to account for those kinds of issues?
Here there are some stuff that we don't know, because the only data we are using is from this site. And this might be limited, and there might be more data available. So I don't know which institution. And actually, for example, in Italy, we have no access to quite good amount of data for specific region of Italy, which was not on this side. So that might be the first way to solve this problem. Then for, the countries that are not doing subsequencing, then of course this algorithm can be valid if you have enough data to make it work. It's something that we are trying to discuss a bit with other collaborators, how we can obtain a more efficient program that work with less data. For now, we don't have a definite answer to how to do that. But it's clear that the Omicron seems to emerge from South Africa, and they don't have so many sequencing available. One thing that, for example, what happened in India is that the Delta variant started there, and very soon went to England. And in England, in the UK, we were able to detect it. So you're just adding a delay, because we could always say that we found a variant that is dangerous in the UK. It's already spreading in India. You're gonna say maybe too late for Indian people, but at least for the UK, you can say it.
Thank you. Martin, you there?
Sorry, I should take my hand down. I mean, it's going to be a problem going forward. Generally, it's kind of like the mismatch with sequencing with a lot of this kind of stuff is going to be a huge issue going forward with labeling variants of concern, or even knowing that they're happening in some places, because the data is just not. Yeah, it's going to be a huge problem for everyone in the field.
Yeah, that's for sure.
Do we have more questions for Sharon? Questions? Anyone?
So if I just might add, so this is really on the early stages of this algorithm. We have it since like a few months, but I mean we are not epidemiologists by all time. So we worked as much as we can, and we have new collaborators each time that join us and help us a lot to improve it. I think it might be able to do a lot better. The sequencing is an issue that, the number of sequencing is an issue that need to be taken into account, but maybe there are different ways to refine a bit the analysis, maybe wait in some way the sequencing that we are not waiting with the Levenshtein measure, but maybe we can find a way to some cases to wait to be able to detect more closely. Second thing is that I'm also saying that the sequencing is not very good, but in the data from this side, we're throwing away a lot of the sequences because they contain some mistake, if I can say. Sometimes the amino acid is not well defined. They put an X to tell us that they don't know what kind of amino acid. And sometimes the amount of data we are throwing away because this I will say bad writing of the sequencing is huge. It can be of order 50%. So if we find a way to, we need to find a way to still use this data without completely a part of this data that will also give us a higher performance for the program. And also the next step also is maybe to use it on other disease than COVID, because I'm saying COVID, but here we are just comparing the sequence of the spike protein of virus. And for influenza, we can do the same. So it's something that we would like to do.
Great, great. Martin, do you want to say something?
Yeah, unless anyone's got another question, because I've already gone.
Pardon me?
Anybody? Okay. In which case, I will. It's just going back a few slides. You mentioned the
Yes.
methodology for going through the sequence stuff. How are you doing? How are you detecting differences between synonymous and non-synonymous substitutions?
Sorry, could you repeat?
Okay, so in terms of genetics, you have 3 nucleotide sequences that code an amino acid.
Yes, yes.
But some of the changes that can occur in this 3 nucleotide sequence still lead to the same amino acid being coded in protein. Is the algorithm that you're using accounting for that?
No, because we only using the sequence of the spike protein in terms of amino acid.
Amino acid, not nucleotide sequence.
Yeah.
Okay, good.
If I said nucleotide, I'm sorry, I misspoke. We could use it on the entire DNA and use nucleotide, but we just restrain ourselves to only the spike and directly to the amino acid.
Okay.
It's true that, I mean, you're right, there are some change that could be maybe maintained through different through the reproduction life of viruses, and maybe we could detect them at the level of nucleotides and not at the level of amino acid. So that's it's a good point.
I mean, if you were going to nucleotide sequence, you would have to somehow account for synonymous and non-synonymous substitutions.
Yes, that is true. That is true. We actually we have a collaborator that wanted us to do this analysis on this on the entire genome, but it's taking much more time. The genome is much bigger, and computing the distance takes more time. But there is so many sequences we have so many sequences, so many distance that's getting too high.
Wouldn't it be better, instead of going for the whole genome, prioritizing the areas where the genome is changing the most? Until you get the whole genome.
But like that, I will say yes. But I mean, we need to know in advance where it's what's changing the most, and for that, I guess we still have to run an analysis unless there are some biologic reason to look on specific place. We still need to run an analysis on the entire genome to see where are the hotspots of the genome, if I can say, and then we will be able to have a more efficient program on just zooming on those hotspots. Yeah, that could be done.
I mean, actually, you could probably just look in the literature, and it would say, I'm sure the analysis would like there'll be a review somewhere saying what's the areas that's changed the most.
Yeah, you're right. We didn't think about using the genome and just zooming on some parts. I mean, this is a good idea. I would like to talk with my collaborators about that. Yeah, very good point. Thank you.
I think it's a great place to stop. And I would like to say thank you so much, Sharam. This is a great presentation, and we are grateful and happy to have you here. And